All right, so we're all done. Unit 2, with the exception of our research project that we still have to do, but that's going to be started next week. And then, like, I think I have 1 day with the computers next week and 1 day with the computers the week after that. So it's not going to be due for a while, like 2 weeks after our last research day. So that's a long way off yet. We'll actually be starting the next unit, which is physics, tomorrow. Obviously, none of what we take after today will be on the Unit 2 exam. Okay? People sometimes get confused with that. Okay? Only biology on the Unit 2 exam, nothing that I teach in physics tomorrow and Friday and whatever is going to be on there. Okay? So what we're going to do is the same thing we did for chemistry. I'm going to go over the key parts, key points, okay, of this unit. I will be posting, as soon as I have this recording done, the review package. So it'll be just like what you got for chemistry. There'll be, a page of kind of review activities and sample questions, and then there'll be a practice test. Okay, so same as you got for that. This will probably be the last time you get that much stuff, okay, for the unit exam, but at least you can take it home and practice and kind of see how I would word things and stuff like that. All right, so you'll have all of that after class today. All right, so the first thing we went over, cell theory. Okay. Should you know the points of the theory?
Yes.
Yes, definitely. Right? Especially since there's 3 of them. It's not that hard to remember. I'm not going to burn a multiple choice question on that. Or sorry, a written response question on that, but you could certainly expect to get a multiple choice question about the 3 points of the cell theory, like which of the following is not part of the cell theory, since most multiple choice questions have 4 options. Okay. Do you need to know the people?
Yes.
Yes. I mean, there's only 4. Okay, so it's not too taxing to know Hooke, Leeuwenhoek, and Schleiden and Schwann. Okay, those are your 4 people and their contributions. Hooke obviously viewed cells first. Leeuwenhoek viewed living cells first. Okay, and then Schleiden and Schwann came up with the cell theory after discovering all organisms were made of cells. Okay, so that's going to be important stuff, but it's almost all multiple choice, with the exception of the second point. What's the second point of the cell theory?
Cells carry out basic functions for organisms.
Cells carry out the basic functions of an organism. Is that something you could work into a lot of questions about cells in the written response? Probably. Yes. Okay, definitely, as opposed to probably. Okay, there's going to be ways for you to work that in. Okay, and that just shows kind of a well-rounded application of what you know about the cell theory. Okay, you got a question that's about cell transport. You could talk about how transporting materials by diffusion is a basic function, and it's done by the cytoplasm, or something like that. A little blurb like that goes a long way, okay, in answering a question about intracellular transport. All right, so keep that one in mind. That's one that could come up, or you could have the opportunity to talk about on more than one occasion in the written response. Okay, second thing we went over was microscopy techniques. Obviously, there's not really a way for me to test your proficiency with the microscope on the test, but I could definitely put a picture on the test and ask you to estimate the size of the objects. And I will. Okay. Here's how that'll look. I'll just have a picture, and then I'll have a circle on the picture. Okay, maybe it's of an amoeba. Okay, big random blob. Okay, and then I say, you're viewing this on high power where the field of view is 400 micrometers. Which of the following would be the best estimate of the size of the amoeba? And then there'll be 4 options, and you have to pick the best one.
So you'll tell us what the magnification is for high power?
Yes, I will tell you. It's on high power, and here's the field of view. You're not going to have to memorize the field of view, okay, for those lenses. Yeah. Okay, so definitely going to be asked to do that. And as I was saying yesterday when we went over the cell anatomy lab, that's still giving people some trouble. So if that's still giving you trouble, make sure you come and see me so we can go through it. All right, there'll also be probably a few multiple choice questions on microscopy techniques, like kind of random things like what magnification do you start on, or things like that. How do you carry a microscope, stuff like that. Okay, and then we went over modes of nutrition and how they relate to cell structure, okay, which was part of our cell anatomy lab that we again went over yesterday. So you definitely need to know these two terms and be able to use them properly. Chemoheterotroph and photoautotroph. I don't know how many times, I can't remember if it was this class or the other class or both, where I had people write photoheterotroph, okay? I don't know what that is, right? It's not a mode of nutrition because then they're light other feeders. So I don't know, they like getting a tan while eating other organisms. I don't know, but there isn't one of those, okay? So photoautotroph, light self-feeding, okay? Chemoheterotroph, okay? Chemicals from other organisms. Make sure you have those straight and can identify them in a multiple choice question or possibly use those terms in a written response item as well, okay? When we talked about modes of nutrition and cell structure, okay, we talked about how there's a lot of similarities, way more similarities than there are differences between plants and animals, but all the differences have to do with their mode of nutrition. Okay, those differences quickly summarized. Plant cells have chloroplasts, a water vacuole, and a cell wall. Okay, which plant cells or animal cells do not. Right? The water vacuole and the cell wall help to keep the cell rigid, which supports the plant overall. And then obviously the chloroplasts carry out photosynthesis, okay, which is their primary food source. And then for an animal cell, no cell wall. They've got to be flexible so the animal can move. Okay? Lysosomes to help digest food, and centrioles to help with cell division. Because the cells are flexible, they could radically change shape and possibly have DNA get lost in the shuffle. Okay? That's why the centrioles are in animal cells and not plants. Everybody with me there? Okay? Those differences, those are important. There's going to be multiple choice questions about why there's differences in cell structure, and possibly you'd have to explain that in a written response item as well. Okay, so make sure you know the modes of nutrition, why and how they affect the cell structure. Should you expect to have to label a cell on the unit exam? Just like what was on your quiz. You gotta give me the structure and the function. Okay, not of everything, right? Probably be like 5 or 6 instead of 13. Okay, but you don't know which 5 or 6 I'm gonna ask about. Okay, so you gotta know them all. Right, but there will be a diagram of a cell, and you will have to identify the structure and give its function if it's labeled on there. Okay. Also, don't forget about this term, compartmentalization, right? And eukaryotic versus prokaryotic. Eukaryotic cells have compartmentalization, that is, they have a nucleus and other membrane enclosed organelles. They're the only kinds of cells that we dealt with. Okay? Prokaryotic cells are bacteria. They don't have any membrane enclosed organelles. As long as you know the difference, that's the big thing. Okay? Eukaryotes have compartmentalization, which means membrane enclosed organelles. Okay? Especially a nucleus. Hey, don't be afraid to ask questions as we go. Yeah.
So like what you mean by it's not like membrane enclosed, like for a prokaryotic, so like a mitochondria would not like have a double cell membrane?
There wouldn't be a mitochondria.
Oh, okay.
Okay, like a prokaryotic cell has no organelles.
Oh, okay.
Okay, all of the processes go on all in the same space. Okay, there's what they call a nucleoid region, but it's just where the DNA is. It's not enclosed, it's not protected. So food gets digested and cellular respiration occurs and wastes are processed in the same space the DNA is kept. Okay,
Okay.
it's like, see, I'm trying to think what it's like. It's like washing your dishes and your clothes in the same water.
Okay.
Okay, if you just made spaghetti, you don't want to use that water to wash your whites. If you haven't done laundry, don't do that, okay? That's how you get like, you know, pink whites, okay? You don't want that. So yeah, it's doing everything in the same space, not efficient.
So like the only thing we would have to know about that is just like one is the, like.
One has it, the other doesn't.
Okay, that's it.
Yeah, that's all you have to know at this level for prokaryotes and eukaryotes. All right, cell transports. Okay, the big three, super important. There will be one written response item. Okay, there will be 3 written response items on cellular transport. Okay, not necessarily one on each type, but that's possible and likely. But you do need to know the big 3, and you need to know them well enough that you would be able to identify what transport type is happening and then explain how it works. Okay, so if I said a bunch of slugs go on holidays to the Salt Lake, the Great Salt Lake in Utah, okay, explain why this would be a disastrous vacation. Okay, using what you know of cellular transport. Okay, what cell transport process would be happening? Osmosis, because it has to do with salts. Okay, if salts is mentioned, then yeah, it's going to be osmosis. Okay, and then you would explain to me. Osmosis is a cellular transport process that moves water only across a membrane in order to balance salt concentrations. In this situation, osmosis would result in the slug shriveling up and dying because water would be drawn out of the slugs and into the salt water, okay, killing them. All right, that's a thorough explanation. It tells me I know it's osmosis, here's how osmosis works, and here's how it's going to work in this situation. Okay, that's what would be expected of you in 3 different questions in the written response. Be able to identify what kind is happening, explain to me how it works, and then apply it to that particular situation. So, diffusion. We talked a lot about diffusion because it's the most important. Okay, what does it transport? Everything. Okay, and where does it transport it? Inside the cell, from place to place within the cell. Okay, it's not about across the membrane. So if the question is asking about a process that's across the membrane, diffusion is out. But it could still be both of the other kind. Okay, osmosis and active transport both go across a membrane. These are the little key kind of characteristics you need to watch for in the question. Okay, so diffusion transports from high to low. Okay, is it passive or active? Passive. Okay, is it targeted?
No.
No. Okay, just goes from high to low. Okay, and it's slow if the distances are very large. Okay, all of that kind of stuff. Those are things you're going to need to know. Okay, we also talked about it in terms of cell size. We did that activity with the cubes. Okay, so we need to know how cell size affects diffusion and vice versa. Why a cell can't get really big. Be able to explain that. That's actually, I have that again here later. Okay, and then osmosis. Okay, we talked about that. You know, if the slug's going to the Salt Lake, you'd be able to identify it. Okay, and then obviously active transport. Active transport is across the membrane. It's usually larger, okay, like solutes, okay, not larger food molecules because that would be phagocytosis. Okay, but it's usually stuff that also needs to be moved quickly, and it needs to be put into higher concentration on one side than the other. Okay, so if something is mentioning about differing or changing the concentrations to higher, that's got to be an active process. Okay, and so then you would identify it as active transport, tell me that it uses energy. The energy affects the carrier or transport proteins. That's a key part of that explanation. Okay, it's their change in shape that causes the pumping action that moves the stuff across the membrane and makes it higher in concentration on one side than the other. Okay, and then we talked about multicellularity. Okay, the key defining point of multicellularity is specialization or division of labor. Okay. In a true multicellular organism, some groups of cells do a certain job, other groups of cells do other jobs. Okay, they do them very well. right? That's specialization. What was our example of a multicellular organism?
Plants.
Plants, okay? In particular, what part of the plant? The leaf, right? The layers of the leaf were our example of specialization within a multicellular organism. Okay, we talked about how the epidermal layers secrete the waxy cuticle, how the palisade layer carries out photosynthesis, the spongy layer aids with the exchange of water, carbon dioxide, and oxygen. Okay, the stomates, which are made of guard cells, can open and close because the guard cells are specialized to change their shape due to osmotic pressure. Okay, stuff like that. Okay, that is definitely something you need to know. I guarantee you there will be a question about that. Okay, so know the layers of the leaf and know why that is an example of specialization within a multicellular organism. Okay, the other thing about multicellularity, okay, its advantage, surface area to volume ratio. Okay, it allows an organism to become very large without having to have that problem with diffusion not working over large distances. Okay, and then this was our examples of specialization. Sorry, I got ahead of myself. I already talked about that. Okay, our example of specialization is plants, the layers of the leaf in particular. Okay, make sure you know what each part of the leaf does, okay, because that's important for that part of the quiz. Okay, no transport in plants. Okay, be able to explain in detail how both water and sugar are moved in the plant. What tubes move them, what direction they move them in, and what forces are involved. And there may be an application question as well. And it could be very similar to something in the analysis for the plant transport lab. Okay, so what I mean is I give you a situation that a plant finds itself in, and you need to tell me how that situation would affect its ability to transport water. Okay, if I have a question that's strictly recall in the written response about plant transport, and it asks something like this, identify the tissues that move water and sugar and explain how they work, okay? What would I be looking for you to tell me about? Okay, I'd be looking for, yeah, osmosis as the force that drives or that pulls water up the plant. What else? Gravity is the force that brings sugars down the plant. Phloem and xylem. Okay. Which one does the phloem carry? Sugar. Which one does the xylem carry? Water. Okay, there's one thing that's missing, because it would be a 6-mark question. Okay, one thing that's missing. We've talked about osmosis helping. What else helps? Hydrogen bonds, polar nature of water. Okay, those would be the things. That's what I mean. Explain in detail. If you think it applies, tell me about it. All right. I can't give you marks for what you know, but don't write down on paper. Okay. I know that's not fair, but it's true. Okay. If you don't put it on paper, then I have to assume you don't know it. So make sure you tell me all the details. Okay. I was telling my period 4 class about this yesterday when we went over their cell anatomy labs. I didn't tell you guys as much because you generally do a pretty good job of it, but the big thing here is level of detail. Okay? Level of detail is the difference between 3 out of 5 and 5 out of 5. Okay? 3 out of 5, you know the basics. You can explain to me how a plant transports water, okay? Or that it's in the xylem. Okay. But when you don't tell me it's osmosis and the polar nature of water and hydrogen bonds make it stick to the inside of the xylem, blah, 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 that's the difference between 3 and 5 out of 5. Okay? Level of detail. All right. Parts of a plant. Phloem and xylem, okay, gotta know those, okay, where, what they transport, forces, so that's what we just talked about, okay.
What do you mean by forces?
Like osmosis,
Oh,
gravity,
osmosis.
yeah, the things that do the pulling.
Okay.
Yeah. Okay, also know roots and leaves and stem, okay, are the parts of the plants. Definitely there could be multiple choice questions about, the leaf and the stem and the roots and what they do. All right, plant growth and development, okay, so you need to know about how tropisms work, you need to know the different hormones that are involved, okay, and you need to be able to apply that to any given situation, okay. So it's hormones and tropisms that allow a plant to adapt to its environment. It's why a plant's final shape is not genetically predetermined, okay? It's an interaction between the plant's environment and the plant itself. All right, so there'll be a number of multiple choice questions about this topic with pictures, all right? And they'd be, here's a picture of this plant, what hormone is responsible for this, okay, kind of thing. So a lot of this is application, it's situational application. Here's a situation, tell me what you know about it, apply what you know to this situation, explain, okay, stuff like that. So there'll be a lot of that. Also, there will be questions in the written response, especially related to tropisms. Okay, I think I told you guys this yesterday where I show you a picture of a plant and then I ask you, okay, what tropism is going on here and explain how that tropism works. All right, now the explain how tropisms work part, is that the same no matter what tropism it is? Okay, remember, a tropism is a bending of the plant, okay? No matter what caused the plant to want to do that, the mechanism by which it makes itself bend is the same. The stimulus can be different. Thigmotropism, touch, geotropism, gravity, phototropism, light, okay? But in any of those three cases, the way the plant makes itself bend is by doing what? Auxins, right? Okay? It secretes auxins to one side. That side grows faster or longer than the other side, and that causes the plant to bend. That's the mechanism. That has to be part of your explanation for a tropism question. Okay? Yes, certainly identifying whether it's geo, thigmo, or photo is important, and then being able to tell me that photo is light or geo is gravity or whatever, yes, that would be like 2 marks. Okay? But the other 3 marks in a 5-mark question about tropisms would be explaining the mechanism by which tropisms happen. Okay? Differential rate of growth caused by auxins on one side more than the other. Okay? Got to be able to explain that. Okay. Factors affecting plant growth, biotic, abiotic, expect a few multiple choice questions on that. around that. I briefly mentioned that before, but I want in particular to say it again. Okay, understand how surface area to volume ratios affect diffusion. Okay, why cells can't get big. Okay, so details. There are 26 multiple choice questions. It's actually less than your chem exam, 7 less than your chem exam. Okay, and as I was saying, the multiple choice questions test stuff about microscopy, cell structure, cell transport. There's a lot of stuff about like cell structure and functioning. There's some stuff about plant structure and transport in the multiple choice. Okay. Multicellularity, people. Okay, so like Hook and Schleiden, Schwann, and those guys. Okay. Prokaryotes and eukaryotes. And more cell structure and functioning, plant transport, I think I said that already. Plant growth and development, and there's a few multiple choice questions about plant growth and development and hormones and stuff like that. And some stuff on microscopy. Okay. In the written response, there are 53 marks. And those come from 9 questions. Okay? The questions in the written response are going to ask about the following things. How plants regulate water loss. Okay, tropisms. Okay, I told you I'd have a picture of a plant and I'd explain the situation it was in and you would have to tell me what's going on there. Okay, labeling and giving the function of cellular structure. Multicellularity. Plant transport. Three questions on cellular transport. That's the big 3. One of those 3 questions also involves cell size, surface area. And then one about plant growth and development. Okay, couple of things to keep in mind for the test. Okay, obviously, level of detail is important, but don't go overboard. And by that I mean this. If I give you a picture of a plant, okay, and I tell you, here's the situation it's in, identify which tropism is responsible, how many tropisms should you pick? 1, okay? If you say, well, it's this one and could possibly have a little of that one, you don't have a clue which one it is. Okay? That's what that says to me anyway. Okay? This is not politics. You cannot sit on both sides of the fence. You have to pick one, okay, and go with it. All right? You have to decide it's this one and explain to me how it works. Remember, on that question, 3 out of the 5 marks are how a tropism works, and it doesn't matter which one you pick, you can get those 3 marks by explaining how any of the tropisms work. All right? The other 2 would be, okay, you identified which one it was, photo, geo, or thigmo. Okay? So just keep that in mind. Okay? You can't just write everything you know for a question and hope that some of it is right. Okay? Because that, well, interesting. It does not tell me whether you knew how to answer that question. It just tells me you know a lot, but not for that question. Okay, so make sure you're writing an answer that has a lot of detail related to what the question is asking. Okay. Studying for this test is a lot different than studying for your chemistry exam. Okay. For your chemistry exam, you practice stuff. You practice balancing equations. You practice predicting reactions. You practice mole equation calculations. But for this one, this is a lot of reading and making notes, and then also kind of coming up with situations where the stuff you know could be applied. Okay? Because a lot of biology is read and regurgitate and read and apply. Okay? And there's a lot of application on this test. Here's a situation, tell me what you know and apply what you know to it. Okay? There's a lot of that on this test, and the way you prepare for that is to read your notes, make some jot notes of your own, try and come up with situations that might apply to that, okay, and then explain it. Right? That's the way to kind of prepare for a biology exam. But it's a lot more reading than it is practicing. Okay? There's really nothing to practice other than how to estimate the size of an object, okay, under a microscope. The rest of it is just reading and knowing your stuff. Okay? How long should you sit and study at one time? Yeah, between 15 and 25 minutes. Okay? And that just has to do with the way your short-term memory works. If you just read and read and read and read and read and read and read for 4 hours, you'll remember the first 3 things you read, the last 3 things you read, and forget everything in the middle, and it'll be a total waste of your time. Okay? What you need to do is study in short chunks, okay, and then take breaks. Allow your brain to process that stuff. You can think about it a little bit while you're on your break, okay, and then go back to it for a while. But just cramming for hours is not an effective method of studying, okay? Study a bit, take breaks. Study a bit, take breaks, okay? That's going to be your best way, especially for something like biology where you just have to put a lot of stuff in the brain, okay? Practicing you could do bigger, longer chunks for, okay? But for reading and remembering, okay, you're committing stuff from short-term memory to long-term memory, okay? Making small chunks is a better strategy there. Okay, what do you need to bring to this test? Something to write with. That's it, okay? Yes, you have a size estimation, but you don't need a calculator for that, okay?
Do you know what day it will be on yet?
Yes,
Next Wednesday.
November 10th, a week from today. Yeah. All right. Yeah, so you just need writing utensils for this one, okay? I mean, you're going to put your phone in the bin, same as you did for the last one. You'll have a multiple choice form and the written response part of the test, and we'll go from there. Yes, you can start early, just like the last time. Okay, we'll say come here. What did we say last time? Was it 10 after 8 or quarter after 8? 8:15. Okay, so have you start at 8. You can come in and start at 8:15 if you want. If you're not wanting to start early, don't come here. Okay, you can't sit in here and visit while people are writing their test. Okay, so yeah, you can start early, but you can't go late because obviously we only have 4-minute breaks and you have a class period too. Okay. All right, that's all I think I have to tell you about it. questions from you guys? Anything you want me to review in more detail?
I just want to kind of confirm. So, like, with like the tropisms and stuff, and then like the hormones with it, the only hormone that affects tropism is auxin.
It is the primary one, so it's the one you would mention.
Okay,
Yeah.
and then
It's not that the other ones don't have a role to play, but it's minor.
so then what do the other like hormones have? Like, where would you find that in a situation, or like they
In a
on
multiple
their
choice
own?
question, let's say I show you, a picture of, let's say, fruit in various stage of ripening. And I ask you which hormone is primarily responsible for this. Auxins, ethylene, abscisic acid, or cytokinins.
Okay.
That would be an application. Okay, other questions there, guys? Okay.
For the cell structure, for the plant one, what is the function of the tonoplast?
The tonoplast is just the special membrane that encloses the water vacuole. So it's especially like elastic and also especially good at osmosis, moving the water in and out. Yep. Okay, what else, guys? Okay, I am going to post the recording then along with the practice materials, the review materials, and you will have the next half an hour to do some review. If more questions come up, then you come up and ask me, and I'm happy to go over them. Okay, but that's what we're using the rest of class for today.